Hello and welcome to the VJOncology Podcast, your go-to source for cutting-edge oncology trial updates. Today we're unlocking the most impactful lung cancer findings from ESMO 2025. We'll dive into 4 key interviews, each of which signals a shift in how we treat or understand lung cancer. Let's get started. This podcast is available on the Lung Cancer channel on VJOncology, an independent medical education platform which is supported by Takeda. Supporters have no influence over the production of content. Our first stop is the OptiTROP-Lung04 trial of sacituzumab tirumotecan in patients with EGFR-mutant non-small cell lung cancer who progressed on EGFR TKIs. In this interview, Dr. Antonio Pissarro describes how this antibody-drug conjugate significantly improved progression-free and overall survival versus platinum-based chemotherapy with a manageable safety profile.
So, the management of patients with EGFR-mutant non-small cell lung cancer that progress to the first-line setting with the TKI alone, generally osimertinib worldwide, is very difficult because the rest instances are heterogeneous polyclonal. At the present time, we know that a target single alteration like MET is effective, but is not so easy to extend on a wide patient population. And in this particular field, we have already approved in the last 2 years different biomarker diagnostic strategies. The first is the combination of chemotherapy plus amivantamab, the MARIPOSA-2 study. The second is the combination chemotherapy plus ivonescimab for the HARMONY trial that is approved exclusively in China. During the ESMO 2025, we presented very interesting data, the OptiTROP-Lung04 trial. This is the first Phase III trial that reported data with an ADC in patients with EGFR alteration in the first trial with the positive results. Indeed, the authors reported that patients randomized to receive a platinum chemotherapy or sac-TNT, and the results showed a very impressive results in favor of all the efficacy endpoints. So we have an improving progression-free survival, an improving overall survival, an improving overall response rate for patients that were randomized to receive sac-TNT. Interestingly, globally, toxicity were good, more or less manageable, and with less grade 3 than the standard chemotherapy. And the only one toxicity with some concern is stomatitis. That is one of the characterizing toxicity for ADC in this kind of setting. Globally, I think that this is a very clear and positive study, developed actually exclusively in China. And now it's important to understand also the anti-regulatory approval to consider if this kind of drugs could be applied and approved in Europe and FDA based on this trial. But it is important to know that at the present time there is an ongoing trial, the TROPHUS-009, that is the global trial with the same study design, sac-TNT versus platinum pemetrexed in patient progression after EGFR tyrosine kinase inhibitors. Interestingly, in this field we have many ADC in the evaluation. The other are datoderuxtecan, ongoing in a Phase III trial, the TL15, TROPION-Lung15, and then we have also isapren, a bispecific ADC against HER3 and EGFR, and TEMA-BA, a very interesting ADC target c-MET with an important way and pathway for this kind of patient. Globally, we can consider that in the last 2 years, with the improvement in the first-line setting, with the combo FLAURA2 and MARIPOSA, and the improvement in second-line setting with the MARIPOSA-2, HARMONY, also OptiTROP-Lung04, the treatment scenario for patients with EGFR alteration is improving step by step, hoping for a better survival for our patient.
Next, we shift to small cell lung cancer. In the Delphi 304 study, tarlatamab, a DLL3-targeted bispecific T-cell engager, was compared head-to-head with chemotherapy. Dr. Martin Schuler reports improved overall and progression-free survival, fewer severe adverse events, and manageable cytokine release/neurotoxic events.
So the Delphi 304 is the first Phase III study to demonstrate superiority of tarlatamab over 3 choices of standard chemotherapy for second-line treatment of patients with small cell lung cancer. The study demonstrated a clear survival benefit and also a benefit of progression-free survival with a risk reduction for death of 40%. Now, in today's ESMO, we are presenting a detailed safety analysis focusing on the overall safety events and then also on the tarlatamab-specific events. So overall, there was less grade 3 or higher adverse events with tarlatamab as compared to chemotherapy. As expected with tarlatamab, there were more cytokine release syndrome events, which are largely confined to cycle 1. And with chemotherapy, as expected, there were hematotoxic events, which are maintained throughout the study. So in the tarlatamab specific toxicities, the cytokine release syndrome, as I said, occurred early in the first 2 injections, cycle 1, and it was mild to moderate in the majority of cases and well manageable. There were only a few rate of dose adjustments or treatment adjustments, dose adjustments and schedule adjustments. I'm taking a short break because you can cut that with a cytokine release syndrome. So the other immune-mediated toxicity of note is the ICANS, which is very, very few events with tarlatamab. There were only a few % of patients experience them, in particular 15 patients, and in 14 of them it coincided with CRS events. And 93% of those fully resolved, and there were basically mild to moderate. The most important neurological AE with tarlatamab is dysgeusia, which was reported by 23% of patients. 3/4 were reported mild dysgeusia, and it did not lead to any treatment stop because of this toxicity. So overall, we can summarize that tarlatamab is a safe treatment, a feasible treatment. It's less toxic than the standard of care chemotherapy, and it's a new standard for second-line treatment in small cell lung cancer.
Now turning to another mutation-defined subgroup, HER2-mutant non-small cell lung cancer. In Beamion LUNG-1, zongertinib was evaluated in the first-line setting. Dr. Sanjay Popat describes a response rate of 77% and disease control in over 90% of patients with favourable safety.
So at the ESMO 25 Congress, I was delighted to present the efficacy and safety data of zongertinib in HER2-mutant advanced non-small cell lung cancer. Zongertinib, as you remember, is an irreversible TKI that inhibits HER2 and is actually quite sparing of EGFRs, thereby limiting and minimizing the EGFR-associated toxicities. It's been evaluated in the ongoing Phase I trial with dose escalation, and the optimized dose moving forward is 120 milligrams once daily. I presented results of cohort 2, which is patients that were untreated previously who had metastatic HER2 tyrosine kinase domain mutant non-small cell lung cancer. This is an important group of patients because they have no targeted therapies approved for use, and the standard of care is currently chemotherapy with or without immunotherapy. A total of 74 patients were enrolled on the study, and the primary endpoint was BICR-confirmed objective response rate. And indeed, the primary endpoint was met with a response rate of 77%, with over 90% of patients achieving disease control. When we looked at the waterfall plot, every single patient had some form of tumour shrinkage, and the clinical benefit of tumour shrinkage was not restricted to whether the patients had a YVMA mutation or not. When we looked at time to event endpoints, median duration of response had not been met yet, and neither had median progression-free survival. So we need more mature data for that. And the toxicity profile was very similar to that previously reported, with mostly grade 1 or grade 2 adverse events.
Finally, we step back from therapies and look at biomarkers. The immunodriver 2 analysis reported by Dr. Jay Lee correlated CD8 T-cell infiltration and PD-L1 expression with survival in first-line non-small cell lung cancer. In this cohort of over 1,300 patients, those with high CD8 and high PD-L1 had not yet reached median survival, while CD8 low and PD-L1 negative had a median overall survival of 11 months.
Immunodriver 2 study is a collaborative study that UCLA did with Tempus, using the Tempus Lens database. And in Immunodriver 2 study, we looked at response median overall survival rates for patients that receive first-line chemo IO or IO alone in the metastatic setting. And what we were correlating was the co-expression of PD-L1 expression with CD8 T cells. PD-L1 expression needs to be interpreted in the context of CD8 T cells, but there is the concept of constitutive PD-L1 and inducible PD-L1. Inducible PD-L1 is where when a CD8 T cell comes into the tumor microenvironment, the tumor cell upregulates PD-L1 to shut down the CD8 T cell activity. And this is why this upregulation of PD-L1 is called inducible PD-L1. And this is most predictive of checkpoint inhibitor efficacy, because coming in with a PD-1, PD-L1 inhibitor will release that brake in that setting. In contrast, constitutive PD-L1 is where there's a baseline expression of PD-L1 independent of CD8 T cells. There could be no CD8 T cells or low-level CD8 T cells, and there's constitutive levels of PD-L1 expression. And this constitutive PD-L1 expression does not predict immune checkpoint inhibitor efficacy. So in the current Phase III registrational trials, we don't look at the CD8 component when we interpret PD-L1 to be, high at 50% or 1 to 49 and less than 1. We only talk about PD-L1 expression outside of the context of CD8 T cells. And I think this is fundamentally one of the problems of why we're seeing heterogeneity and readouts of trials where PD-L1 has not been performing consistently, because it's by random chance how much inducible versus constitutive PD-L1 is present. So ImmunoDriver 2 looks at the co-expression of the two together. And we separated patients by PD-L1 high, 1 to 49, PD-1 negative. And then we looked at CD8 T cells at a median cutoff. And if it was above, we grouped them as CD8 T cell high. If they were below the median T cell counts, then it was PD-L1 low. And what we found is that if your PD-L1 expression is high at any or progressively increasing from 1% all the way up to 50% and higher, and they were also correlated with CD8 T cell high, those patients performed the best. And the CD8 T cell high and PD-L1 high didn't reach its median overall survival. It was at 22 months and was still not reached. Whereas on the flip side, if you're CD8 T cell low and PD-L1 negative, these patients had the poorest overall survival, median overall survival at 11 months. But all of the other groupings between the two analyses, all of the CD8 T cell high groups all outperformed the PD-L1 varying levels, especially if they're CD8 T cell low. So the take-home message, and this was over 1,300 patients in the first-line setting with chemo IO or IO alone, is that maybe in future trials, we should revisit and go back to the fundamentals of enriching the patients who are most likely to respond. And that's going to be the PD-L1 positive patients with CD8 T cells. And also in the implication in the early stages, following resection, when you have the entire specimen, whether it's in the adjuvant setting or post-chemotherapy, chemo IO, and you have the resection specimen, the tumor microenvironment should be interrogated for, are there CD8 T cells present? And that should be a useful clinical parameter. And we should validate this in Phase III trials of whether those are the responsive patients with more additional immunotherapy in the adjuvant side.
So how do these four highlights tie together? They represent three major themes we're seeing at ESMO 2025 and beyond. New forms of therapies like ADCs and bispecifics, expansion of effective targeted agents into new genetic subgroups like HER2, and smarter patient selection via immune biomarkers. If you're aiming to stay ahead in lung cancer care, you'll want to think in terms of what's next rather than what's current. Thank you for listening to this VJOncology podcast. If you found this episode useful, please share with your peers and subscribe for our next update. Until next time.